Stanford University.
Well, good evening, everyone. Glad to see you're all here tonight. I imagine after last week's presentation on obesity, many of you have been managing your weight more carefully over the last days. You look great, so I know that's been happening. Tonight, you'll likely hear somewhere during the presentation that if you really restrict your caloric intake, you might live longer. I don't know whether that's a good idea or not, but you can make your judgments about that. But when you begin to think about the topic of tonight, which is on aging and indirectly longevity, it begins to bring together some of the issues that we have been talking about during this quarter and indeed antecedent ones. Lots of genetic issues. You know that inherently because you look around in families and you note that some people seem to live longer than others, but there has now been an increasing amount of genetic sophistication about aging as well. And obviously, lots of environmental factors. I mentioned one, nutrition being a factor. Of course, you've heard me talk before about exercise and how important that is. But where you live, what the environmental factors are can play a very important role among other issues as well. So tonight, we're going to do an exploration into aging. It may shock you and amaze you to think that some of the most fundamental findings on aging have actually come from the study in the worm, a simple species. You thought you were complex, right? But you'll be interested to know that worms, the kind used in most studies, have about 18,000 genes, and you've got about 22,000. So don't feel too exuberant. Because there's a lot more complexity, of course, to that. But there's been a lot learned in experimental models. And tonight, we have two wonderful speakers, both pioneers in their own way. Anne Bernet, who will come second, comes to Stanford from actually starting out in France. She went to school in Paris initially, did her PhD at the University of Nice. You might all say it's very nice. If you've ever been there, it is. And then moved to Boston to do her postgraduate training at Harvard in a laboratory that was actually very close, she probably didn't know this, to the building that I was working in during that very same time. She was in the Enders building, I suspect, which is associated with the Children's Hospital, where I was on the faculty as well. So it took us both coming to Stanford to meet together. And her research is really enormously exciting because it's beginning to focus on some of the fundamental issues of aging at the molecular level and addressing the interface between aging and stem cell biology, which is another really important factor in this process. But we're going to start tonight's session with a presentation by Tom Rando, who's our second speaker. We've got another duet, and he approaches this process from a different perspective, although he also has that big H bias by having been an undergraduate and medical student at Harvard, trained on the East Coast initially and then came to UCSF, where he did neurology. You could see these connections, right? They keep coming up over and over again. But we are very careful. We only take the very best after they've had their pre-training and then bring them to Stanford, where they really excel. And Tom has done that and has achieved considerable national recognition for his work, which is on stem cells and particularly muscle stem cells, which has a lot to do with regeneration and rejuvenation. He's been recognized for his pioneering work by being an NIH pioneer. This is a very prestigious award that the NIH gives each year to investigators who are really doing out-of-the-box thinking. They've actually awarded 81 for the entire nation over the last 5 years, and I'm happy to say that we have 15 of them here at Stanford, so a pretty disproportionate share, which only affirms my comments on selectivity. So Tom will not only speak from the perspective of his research work, but also in his capacity of leading the Center on Aging, both at the VA hospital, where his primary labs are, but also as the Associate Director of the Center for Longevity here at Stanford. So Tom, if you could get us started. Okay.
Well, thank you very much, Phil. It's a real honor and pleasure to be here. This sounds like a great forum from what we've heard. Very interactive, and we will welcome questions along the way. I will start my talk with really talking about some very general points about aging and longevity, how we measure it, how we define it, things like that, which I think will be very important. And then get into some of the background on what we think aging is, and finish up, I hope, with some work from our own studies on aging and stem cells. Okay, so we'll start with some definitions, and again, these will become clear. I think there'll be some questions that will arise from these, but we'll talk about longevity a lot tonight. We'll talk about aging quite a bit. And just as a starting point, I would make this distinction that longevity is a measure, something we can measure. It doesn't imply a process, whereas aging is the opposite. Aging is a process that goes on, that can be studied, but is related to longevity primarily by the fact that aging organisms ultimately die. And when we measure longevity, we essentially are measuring how long an organism lives. So I want to make one comment about population aging before I get into the discussion about how individuals or individual animals age. And because we talk a lot in the society about an aging demographic, an aging population. And there are a lot of interesting aspects of what makes a population old and what makes the population of one country older than another country, things like this. But the main point I want to make is that a population can age without any changes in the aging or longevity of its individuals. I mean, for example, if it were the case that suddenly we had a mass immigration to the country or away from the country of young people, that would just change the whole population aging structure and could lead us to be a more youthful society or a more aging society. And this is just an illustration of the population distribution in this country at 1950, 2005, and then projected for 2030. And you have males on one side and females on the other side. And it just shows how our population, the shape of our population is changing from a more pyramidal shape to a more rectangular shape. And this is primarily because of really the movement of baby boomers through the population. So what was a large population of young people becomes a large population of middle-aged people and ultimately a large population of older people. Of course, as that changes, our population will return, perhaps, more to a pyramidal structure. But other countries can look very, very different in terms of how their population looks and whether they're a youthful society or an aging society. Okay, so let's talk about human longevity, or in some cases we'll talk about animal longevity. But I want to begin with these definitions and how do we define it, how do we measure it. I'll then move on to some points about what are the determinants of longevity, what determines how long we live, and then end with some questions about is there anything we can or should do about that. Okay, so how do we define it, how do we measure it? So when you ask a question of how long do people live or how long do mice live, what are we really asking? And it really depends on who's asking it, what they mean, because they can usually mean one of two different things. If we talk about actuarial tables and how long we live as individuals born today, we're usually talking about life expectancy, or the average amount of time that a person born today can be expected to live. And that's determined, and I'll show you this in a graph in a second, by the age at which 50% of the people who are born, say today, are alive. And that becomes the average life expectancy of that cohort of people. Now, by contrast, there's another interesting aspect of the question, how long do we live? And that's how long can we live? And that's what would be considered the maximum lifespan. And that is not something that's so easy to measure because really that's determined by how old the oldest person or oldest mouse or oldest worm ever seen is. So that's a number that can change. Actually, I heard that the most recent oldest living person died yesterday. So there's a woman in Japan from a famous place, Okinawa is where a lot of, she was from Okinawan, 114 plus, almost 115, died yesterday. So there's a new 114-year-old who's the current world record holder. Okay, so this is a graph that you're going to see a lot of curves that look something like this, and it's a so-called life curve, or you heard different names given to it. But basically, the way this presents the life curve of a population of, say, people or mice or worms, where you pick a date, say 1950, and you take 100 people and you ask how many of those 100 people are alive as a function of time since birth. Now, with humans, there's always a little bit of infant mortality, but in a developed society, this is a very typical curve where there's very, very little death through the ages of 50 or so. And then people start to fall off, and then the curve trails off like that. So this is the way we we plot the population, and then from this we can calculate average life expectancy, and we can estimate maximum life expectancy. So just as I said a minute ago, if we just take this curve and we ask, when 50% of the people have died or are still alive, what's that average age? So here it's around 70. So this was, again, a typical life curve for people born around 1950, say, 1940s and 1950s. The average life expectancy was around 70 years old. Now, again, the maximum lifespan is somewhere out here. It's hard to know exactly where that is because this 100 people is certainly not going to reflect the oldest person in the world. And so that number is somewhere out here, but you can get a sense of where that is from where this curve tails off. So one of the things that is a common phrase is to say that we're living longer. Now, what do we mean by that? Do we mean that we're living longer on average, or does it mean that we're living longer in terms of maximum lifespan? So we can get a sense of that by looking at these life curves and looking at the averages across the history and across the time when humans were inhabited the Earth. And we have some evidence of what the population curve looked like. This is 50,000 years ago, and of course this data is really just an estimate based on the fossil record. But the life curve looked very differently 50,000 years ago than today. But that would have been at that point, and then still in the Stone Age, but 15,000 years ago in Europe, there began to be this knee in the curve. And then as we move through time, again, the estimates become a little bit more accurate. But Rome, 1100 BC, so well past the Bronze Age and the Iron Age, again, getting more of a knee in this curve, and then up to this curve in, say, around 1970. So if you look then at the average life expectancy, you can see there has been phenomenal and dramatic changes in average life expectancy over this history. So at this point, in 50,000 years ago, people lived pretty much just long enough to propagate the species. So just long enough to have offspring, and the species was propagating basically just as people were coming to reproductive age. And that's very typical for animals in the wild or for people in a relatively uncivilized situation. Of course, as civilization evolved, more and more people began to survive these early years, and more people could survive into the late years. So there's been a dramatic increase in average life expectancy, and this is just another way to look at this. This is actually looking at the average age of individuals across historic and even some prehistoric times, and you can see this dramatic increase in average life expectancy. And one of the important points is that the average life expectancy doubled over many thousands of years, and then doubled again over the last 100 years. So even over the period of recorded history, the increase in lifespan was increasing gradually, and in the last 100 years, we've had a dramatic increase in that average life expectancy to where we are today. Now, what about maximum lifespan? Has that increased recently over time? We really don't have a good way of knowing, because we obviously have a hard time even today determining with certainty how old people are who are the very oldest old, because birth records are not so accurate, and now we're beginning to keep better records, but it's hard to know what the maximum lifespan would have been during this time. But you can see it clearly has not changed dramatically like the average life expectancy did. And in fact, even from historical records, there are some fairly well-known people whose ages were roughly known. So Sophocles was considered to have lived to be about 90. And Hippocrates was also considered to be a very long-lived individual, between 90 and 100. And then, of course, the world record holder is Methuselah, who lived 969 years, and then he died. So, and this is a famous person who you may have heard of, Jeanne Calment. She's actually the world record holder, as far as we know, the oldest person to have lived with a known birth date known to have lived. She lived 122 years. And there are amazing stories. She lived in Arles in France, and she died in Arles in France. And she became very famous, of course, as she passed her 110th, 115th birthday, and she was always surrounded by journalists. So there are many wonderful anecdotes about her life and her secrets to longevity. She quit smoking at 119. Maybe that was her secret. She did all these great things. She took up fencing in her 90s. She was riding a bike at 100. So really a very vibrant person and a very, very witty woman, even right up until the time she died. There's become an interest in so-called supercentenarians, and these are people who are 110 years or older. And this is just looking across the world at different countries and estimating the number of people who have reached this age of 110 or older, and some of it, again, is not very accurate data, but people are looking more and more now at these populations of individuals who are centenarians and supercentenarians, and of course, a great interest is in the families of these very long-lived individuals, as Phil was mentioning, to try and understand what the genetics might be, as well as the environmental influences that allow some people to live these very, very long lives. So here, that's what we're looking at. These are sort of how we measure it. What actually determines how long we live? So in some ways, if we start thinking about genetics as a determinant of lifespan, I was trying to think about how to phrase this, but I think of this as the big lifespan variation and the small lifespan variation. And by that, I'll show you what I mean. So if we talk about the difference between any one of us living what would be considered an average lifespan and a maximum lifespan, we're talking perhaps a 30 to 50% difference over that period. So living to be 75 or 80, or maybe being lucky and living healthy to 110 or more, that's a pretty wide range in terms of number of years, but a small percent difference in terms of our actual total lifespan. On the other end of the spectrum are the genetics that determine how long individuals in the whole animal kingdom, what their lifespan is. And then we're talking about 5 orders of magnitude difference between the shortest lived species and the longest lived species. So you start looking at some fish and giant tortoises and blue whales that can live up to 150, 200 years, and then down to species like mayflies that live less than a day. What is it that determines that vastness of lifespan difference in terms of the genetics, which can be quite complicated even for some very simple organisms, that is so different than what we talk about in terms of modulating lifespan in terms of living between 70 or 80 or 90 years. So this is the big question about genetics and lifespan, but what we'll focus on more is the, just to superimpose that sort of small variation I talked about in man, it's very small across this logarithmic scale over 5 orders of magnitude. So, of course, what's most interesting to us tonight, I think, is to talk about this difference is what is it about the environment we live in, the genes that we possess, that might determine our likelihood of living to an average life or a maximum lifespan for humans. So, as I mentioned, it's interesting to think about how our life curve differs in society versus in the wild. And this is really a typical life curve, as I mentioned, probably prehistoric humans, but animals in the wild. And in this case, what determines the shape of this curve has a lot to do with the environment. So predation, starvation, exposure. So all of the things that really lead to death in the wild create this life curve, which looks very, very different. But if we look at this, what I call longevity in captivity, with us being in captivity in society, but this is what also the life curve looks like for farm animals or experimental animals in the laboratory or animals in a zoo. So you've eliminated all those early life threats. So we can feed them, we can feed ourselves, we keep our young alive, and then we start to see this a very different life curve. And I think of this as having 2 phases. So there's the drop-off phase, where we're coming along nicely here, and then we start to fall off here, and then there's the tail of this life curve. So what's happening here? What does this have to do with in terms of biology and medicine that we can learn from what these kind of early deaths are in this life curve of humans? Well, clearly these are primarily due to age-related diseases. So this is when, these are from 2001, leading causes of death in the U.S., heart disease, cancer, stroke, and so forth. So these are the diseases to which we become susceptible as we age, and these are the diseases for which age is the most potent risk factor, and people start to die from what we would consider age-related diseases, and it raises the question of how does one distinguish issues of aging from age-related diseases. And this is an important distinction, and people argue about this in the field, is whether aging itself is a disease. But medically, we certainly separate them. We certainly call these diseases. We don't call aging a disease. And these are the causes of what leads to people to die typically in their 50s, 60s, 70s. So what happens out here is a very interesting and different question, because this is something we talk about in a lot of our courses. In fact, Ann and I just came from a class we teach in graduate school on aging and longevity. And this is a topic that we get into some very interesting discussions about, is what does dying from old age even mean? Because if you look at people who quote, die from old age, we don't really have a very good characterization, biologically or medically, as to what the cause of death is. So, and it's very different if you look at people who, quote, die in their sleep, it's unknown why they die. But they're reaching some kind of biological limit. And it's interesting that when we study worms and flies and mice, we measure their longevity by keeping them alive and measuring how long they live. And we don't know why those animals die either, for the most part. So they die. Sometimes they also fall off this curve from cancers and other diseases. But for the most part, we don't understand the biology of death from old age. And we really have to begin to understand that and grapple with that, because there is something, and especially across this enormous spectrum of animals in the animal kingdom, or many orders of magnitude difference, that put this stopping point in very different places. So it's an interesting biology that has really remained, in my opinion, unexplored. So is there anything that we can do about it, about our ability to live a certain length of time? Are there things we should be doing? We obviously talk a lot about diet and exercise, and you'll hear more in the next lecture about things that might be very important in terms of living longer. But here's where we really start talking about aging instead of longevity. So Shakespeare had a rather bleak view, I would say, of aging, and we tend not to think of it as rotting and rotting, but certainly there's a decline, and certainly there's a decline in function. And the question is really whether there's anything we can do about that decline. Are there any ways we can influence our aging process and then ultimately how long we live? So really the fundamental arguments in the field come down to whether we age because of our environment or we age because of our genes. And I'll just touch on this briefly, and Anne will talk about this in more detail. But just to introduce this, in the first case, we're talking about extrinsic determinants. So basically the idea there is that aging is a result of exposure of our cells, our tissues, the cells in our body to environmental toxins of one kind or another. Things that are deleterious to our molecules, our cells, and our tissues. And the analogy that I actually think is really one that is quite apt is it's like rust. Okay, so the humans are really evolutionarily designed to grow, mature, and reproduce. Okay, but when we keep them in captivity, we put them in civilization, we begin to see these other features emerging as we keep people alive longer and longer, or mice or flies. And that what we're seeing is like a contraption that we've built to do one thing, but then we keep it around longer and longer, and you start to see rust. Okay, so what is rust? Well, rust is iron exposed to water and oxygen. Here's all the chemistry if you want to go through it. And through these reactive species, and you end up with rust. Okay, so it's an interesting chemistry, but it's also an interesting biochemistry because it turns out that there's not such a different thing that occurs in our very bodies. So this is a diagram of a mitochondria in a cell, and indeed, one of the characteristic features about mitochondria is they obviously are in an aqueous environment. They use oxygen, and the product of turning oxygen into energy is a byproduct in the presence of a lot of metals like iron and other metals that produce reactive species that are damaging to the cell. So very much like rust, because we live in an oxygen environment and because we're aqueous, we will naturally produce, as a normal byproduct of our metabolism, molecules that are deleterious and that actually damage our DNA, our proteins, and our membranes. So this whole field of so-called oxidative damage is a very, very powerful theory as to why we age. That in fact, since we can't get out of this situation of being aqueous by nature and living in an oxygen environment, we will by nature experience over time the accumulation of damage from the very metabolism that we need to live. Okay, so what about the idea that aging is not just this exposure to the environment, but is in fact genetically programmed? And this is a theory that had been very popular for many years, has been very much challenged by evolutionary biologists, but there was an initial theory of aging that we were actually programmed to die. So that we had a genetic program that would, post-reproduction, eliminate older people from the species. And the argument was sort of an altruistic one, that if we could get rid of old people, there'd be more, and there's limited food supply, that would be for the younger people who would then reproduce. And so that was a theory that had sort of wide acceptance, but fell really in the face of evolutionary theory, that we couldn't have evolved so much to have a genetic program that forced us to age and forced us to die in old age, for the simple reason that through evolutionary time, virtually all of the people who are alive never reached old age. So if people are dying in their teens and 20s, then there would be no way for a genetic program to evolve, for us to have acquired a genetic program that would be determining something that would happen to us at 50, 60, 70 years old. So that idea, that sort of altruistic idea that we have a genetic program that leads us to age and die as the reason why we age, has been, I would say, discredited, although there's no doubt, and this is the interesting thing, there is no doubt that our genetic program influences the way we age, and that even single genes differences, single genetic mutations, can influence the aging process. And this you'll hear a lot more from Anne next. So the question is, are there experiments or experiments of nature that really allow us to understand this balance between how genes influence the aging process and how the environment influences the aging process? So are there environmental influences or genetic changes that either can slow aging and extend lifespan, that can accelerate aging and shorten lifespan, or that can reverse the aging process? So I'll touch on these, each one of these. So can it be slowed, can it be accelerated, and can it be reversed? Interesting idea. And again, this you'll hear more in detail from Dr. Brunet. So the idea here would be, can we actually increase maximum lifespan and get beyond this magical 122 years? And can we influence in any way the environment or genetics to get individuals to live longer? And is there a reason to think that we can actually reach much older ages, 150, 200? And certainly this is it's reached the popular imagination. People really have this idea, and there are groups out there looking for ways to have individuals live longer, and there are people who believe that the first person to live 200 years is alive today and will eventually live 200 years. So that's really quite a paradigm shift. I doubt it. I would bet against that, but there are people who are betting on that. Now, what about instead of, if we think about things that can extend our lifespan, instead of thinking about things that extend our maximum lifespan, what about if we just extend average lifespan? So as I mentioned, average lifespan in this graph is around 70. So even without necessarily changing the age at which the oldest person will be, can we increase the average lifespan of people alive today? And certainly that's what most biomedical science is focused on, is reducing the incidence of the diseases that tend to kill us in this drop-off phase. So it's been referred to in different ways, but what I'm talking about here is what has been called the rectangularization of the life curve. So wouldn't it be interesting if, in fact, there is a sort of biological limit out here, around 120 years, and wouldn't it be interesting if these diseases of aging, heart disease and stroke, if we made tremendous medical progress, and then instead of having a life curve that looked like this, we had one that looked like this? It would be very strange, because we all reach a point and then we would just suddenly die. So unlikely to be true. And actually my feeling is that everything that we do here will shift this gradually. So this curve will always look like this, I think. So we'll never reach this true rectangularization. It's also been called the compression of morbidity, the idea that we have a very young life here with very little morbidity, and most of the morbidity that is experienced is out here. Well, wouldn't it be interesting if that period of morbidity could be compressed to a very, very few years after the age of 120 or something like that? Again, it's a strange concept of how society would be if, in fact, we all lived to a very, very close to the same age and then died. But that's the model. But what we focus on is, in fact, treating these diseases of aging so that we can not only extend lifespan but extend health span. And that is really, obviously, the major goal of biomedical research. Isn't so much to extend the maximum lifespan, but to have people live as long as possible with as little morbidity as possible. So can aging be accelerated? Okay, is there such a thing as accelerated aging due to either a genetic or environmental influence? And indeed, there are single gene mutations that shorten lifespan. Okay, now, but that comes with a bit of a caveat, because obviously one can think of mutations that could occur that just cause a disease. So showing that a gene shortens lifespan is not the same thing as showing accelerated aging. In fact, you can think of some examples of single gene mutations that lead to childhood cancers, familial causes of atherosclerosis. There are many, many, many causes of early death from single gene mutations that don't really talk about accelerated aging. They don't really tell us anything about the normal process of aging. But there are some that still have this caveat we have to be careful of, but are compelling. And I just want to show you a couple of examples of those. So these are these disorders called the progerias. They're also called the segmental progerias. And they're really been thought of, both in humans and now in animal models, for many years as possible examples of true accelerated aging. And so this is a disorder called Hutchinson-Gilford progeria. And it is a disease that occurs from a gene mutation for one protein called lamin, which is in the nucleus of every cell in the body. So single mutations, one gene, one protein, part of one organelle, and you get a very strange phenotype because this individual is actually 15 years old. So this is a disorder that starts in childhood and progresses very rapidly, and the individuals show remarkable signs that are similar to what we think of as people who are quite old. Now, is this accelerated aging? That's always the question that arises in these cases because, in fact, if you look at all of their tissues, they don't really just look like old people. I mean, they appear to be like old people, but if you look across the kinds of diseases they get, the kinds of problems they develop, they're not just a compressed 70 years into 20 years. So they're not quite the same thing as just an accelerated aging, but they are remarkably compelling in terms of appearing to be accelerated aging, and many, many tissues are involved. So here's another disorder called Werner syndrome, and this is from, again, a single gene mutation, and this is a gene that encodes a protein that is involved in DNA repair. Okay, and here, this is, unlike Hutchinson-Gilford, this is a disorder that manifests itself later in life. So here's an individual, and this is the same person years later, and in fact, she's 48 in that later picture. So normal appearance at age 15, at a time when Hutchinson-Gilford is very, very advanced, but then suddenly begins to develop this apparent, very rapid aging syndrome. So again, these progerias have been very powerful, and there are now animal models of these mutations as well as many others that have been used to study the aging process with the thought that if we can understand how these single genes and single proteins might lead to accelerated aging, maybe we could understand then how to either use these same proteins or use these pathways to slow down the aging process. So can aging be reversed? Now there's an interesting question. And one doesn't really get into this very much in terms of the field of aging until one has some surprising results that looks like it's possible, for example, to take a situation where you have an old tissue or an old cell, and you can impose on that the characteristics of a younger cell or younger tissue. And I'm going to share with you some studies from our lab that give a hint at, number one, what the aging process might entail and how that might be modulated experimentally. So as Phil mentioned, we study stem cells, and we actually study primarily stem cells in skeletal muscle. And these are the cells that are responsible for maintaining your muscle. They repair your muscle when it's injured. And these are just adult tissue-specific stem cells, which exist in pretty much almost every tissue in the body. So they're very prominent in terms of tissues that are turned over very rapidly, like the skin or the gut or the blood. They're less frequent in tissues that are very stable. There may be some in the heart. There are certainly some in the brain. But across most of the tissues, the lung, the kidneys, one can find this population of stem cells that maintain and repair that tissue or that organ. So we're interested in skeletal muscle, and we're very interested in the process by which the stem cells in skeletal muscle maintain and repair normal tissue. So this is normal skeletal muscle. As a typical appearing Stanford young student might see walking across the quad. It's contrast. So this is a younger adult, and then, as we know, with age, things change with skeletal muscle. And this might be actually more deconditioning than age-related changes. But with age, one of the most prominent changes that occur in skeletal muscle is atrophy, and actually goes by the name of sarcopenia, which is the age-related loss of skeletal muscle. And it really has a profound impact on individuals with age, to the point where people lose mobility, they lose the ability to stand from a sit and get out of bed. These very, very important day-to-day functions. And I think, interestingly, there's becoming increasing interest from the pharmaceutical industry of this topic, because right now we really have no therapies for age-related muscle atrophy. I think that the success in treating osteoporosis has really stimulated interest from the pharmaceutical companies, because osteoporosis, of course, wasn't always a disease. It was considered to be normal aging. With age, people lost bone mass. And then suddenly we began to do two things. We began to measure it, and we began to treat it. And then it was defined that osteoporosis at some point went from being normal aging to a disease when you cross some threshold. And I think the same thing will happen with skeletal muscle. It will become defined as normal aging, but past some point when it leads to morbidity will be considered a disease, particularly when and if therapeutics are developed. So this is a feature of aging that is somewhat unique to skeletal muscle. It's kind of shrinkage of size. But there are a lot of changes that occur in tissues with age that are very common. And one of the features that one sees, so I just use this slide to illustrate a younger male and an older male. This is, of course, father and son. And that if you look at the tissues of a young person, in response to injury, say a wound, those tissues heal very, very quickly, very effectively, and completely. Whereas the same kind of injury in an older individual, even a healthy older individual, will heal more slowly, less effectively, and usually with some component of a scar tissue. So if you have a skin wound in a young person, they'll heal without almost any evidence of a scar. And the older people get, the more of a scar is formed. So there's less of a regenerative capacity in tissues as we age. And this is true in skin, this is true in gut, this is true in bone. Every tissue that we know of where people have looked carefully at the wound response, it's less effective as we age. So this is just an illustration of an experiment from our lab, just illustrating that point of impaired tissue regeneration with age. So these are from the mouse. This is a young mouse. This is an old mouse, so it would be equivalent to, say, a 70-year-old human, and a very old mouse, say, equivalent to a 100-year-old human. And what we're looking at here is muscle that has been injured and is in the process of repairing. So each one of these structures is a newly formed muscle cell that will fill in, and in between here is some connective tissue that would, if not removed, would form a scar. But this is a few days after the injury into the repair, and this is still being recovering. So this tissue will recover completely normally, but if you look at the same kind of injury in a 24-month-old mouse, you see fewer and smaller of these muscle cells and much more of this connective tissue, and at 32 months, it's even more dramatic. So when this is finally recovered, you'll see some normal muscle, but there'll be a lot of scar tissue formed. Okay, so this is just an illustration. If you looked across tissues with this kind of traumatic injury in the body, you'd see the same kind of age-related decline in the regenerative capacity. So one of our first questions was, do we lose this regenerative capacity because we run out of stem cells? And the answer was an unequivocal no. We have plenty of stem cells, and this mouse has plenty of stem cells even at this advanced age, but they just don't seem to be responding as well or behaving as well as the stem cells from the young animal. So we've studied in detail what's wrong with the stem cells in these old animals. Can we begin to understand biochemically, can we begin to understand molecularly why the cells that are there seem to be responding poorly to this stimulus of an injury? And we've done that in some detail, and we've worked out some molecular pathways that are essential for stem cell function in the young and that are impaired. We see them to be impaired as these mice get older. So our first question was, well, if we understand an important pathway for stem cell function in the young, and we can mimic that in the old, can we kind of wake up these stem cells that are there but not functioning in the old animal? So this is another experiment in which we've done that exact type of intervention. So here we're looking at a small injury. So this is normal skeletal muscle. These are these fully mature, large muscle. We call them fibers. And here's an area of injury. This is a few days after the injury, and this is in an old mouse. So you see a few new muscle fibers being formed, but a lot of this, again, scar tissue in its place. So this is a wound that will not heal very well and is healing very slowly. But if at the time of injury, in another old mouse, we produce the same injury, but we introduce an activator of one of these molecular pathways called the Notch signaling pathway, we see very, very healthy, normal regeneration like we would see in a young mouse. Okay, so one molecule, one pathway, and we can stimulate the stem cells in this old animal to now behave like young stem cells. So in a sense, we've restored the youthful regenerative capacity in this area of this mouse. So this was a profound result to us. This really made us start to think, what is it that is wrong with an aging individual in terms of the stem cells they have in their tissues and the responses of those stem cells? And yes?
Just a question related to the old mouse there.
Yeah.
So, kind of going back to your rust on the car, have you figured out why the old mouse is not able to repair the muscle? And what if those things were improved so that he could repair the muscle as opposed to needing the stem cell?
Okay, so the question was, do we understand by the rust analogy what's wrong with the tissue, and is it possible that, I think you're saying, can we modify the environment so that the muscle repairs itself? Is that what you?
Yeah, kind of. So in other words, if you understood why over time that the ability to repair that muscle decreases, then you could put in place some sort of modifications over time again to say, well, we're going to do this, this, and this, so that the ability doesn't go away.
Okay, so you're right. So the question again is, can we intervene? If we understand what the age-related decline is due to biochemically, say, can we intervene at different times so that we can keep the youthful behavior alive and well, as opposed to seeing that function decline? Can we introduce into the animal or into the tissue something that will allow it to maintain that youthful phenotype? A tune-up. Okay, so keep it. So I'm going to show you an experiment that speaks to that question. And the answer is that's exactly what we're seeking, is to understand molecularly why things change, and then can we reverse that, or if we've already gotten to an old animal, can we reverse it, or in an aging animal, can we stop it? So our question from this experiment was really along the lines of what you're asking, is if it takes just one pathway to be activated to take an old tissue and make it look like a young tissue, what would happen? Yes?
Activate a specific signaling pathway in a specific cell.
How do we do it? So the way we do it is, so all of the stem cells that are sitting in this tissue have a receptor for what's called the Notch pathway. So what we introduce is just, when we produce the injury, we just inject an activator of that receptor. Those cells see that molecule we've introduced, it activates the Notch pathway, and it wakes them up. And in fact, we had previously shown that the problem in the old animal was that the stem cells that have this receptor are just not getting a signal to activate that pathway. So we gave them the signal. Okay, so we're trying to figure out why they don't get the signal, but we gave them the signal, and then they behave like normally, or they behave like young cells. Yes?
Building up the muscle more than you, can, say, an older population do more physical therapy or something? Would that help, or is it just done at a molecular level that they're just not going to be able to recover?
So the question was, can an older population, for example, undergo physical activity to help with this process? We don't know that physical activity enhances muscle repair. But there's no doubt that physical activity, even in older individuals, can slow the progression of muscle loss. Okay, so in one case, muscle loss is just this atrophy that occurs. Here, we're talking about something that's really an injury that has to be repaired. So we don't know whether exercise enhances the ability of an injured tissue to repair, but we certainly know that exercise maintains healthier muscles for a longer period of time. Yes?
Where does the signal normally come from?
Okay.
From surrounding
It comes from surrounding cells. That's a good, so the question is, where does the signal normally come from? So in the young animal, well, I don't have another picture here, but if this were a young animal, which it looks like, and we had produced an injury right here, the signal comes actually from these cells that are uninjured around it. Okay, so there's a lot of this, the normal signal that goes to the stem cell that comes from the adjacent normal tissue. And for some reason, in the old animal, that signal is not being produced or secreted. So we experimentally introduced it here, and basically by that simple technique, restored this youthful property. So our question in a general sense, yep, go ahead.
When you do this repair, is there any side effects such as overgrowth?
So we introduce this molecule one time with one injection, so that we don't see any detriment. We haven't really examined it in detail, but if we look at this response, recovery looks normal. Now, we think that if we continue to introduce this signal repeatedly, it would lead to an adverse effect, because normally in a regenerative response, that signal comes on and then gets shut off again. So we tried to mimic that by doing a single injection at the time of the injury. Yes? Is PRP? Oh, so is that part of, is that stimulating stem cell activity? Actually, so the question is, does PRP enhance stem cell function? Actually, we've never, approached that. I'm not sure that there's any reason to think it would have this kind of effect, but I'm not sure. Okay, so when we saw this result, again, our thinking was we have to understand what it is about the environment of these stem cells that is keeping them from activating normally. So we have some ideas there are signals missing, but they're there. In a sense, they're dormant. So they're capable of normal function if given the right signal. So the question was, can we change the environment, the whole environment, without doing an injection of this one molecule? Can we change the whole environment of the old muscle stem cells so that these cells now reacted as if they were in a young environment? So we did then a very strange experiment, and this is called parabiotic pairing. So in this case, we can take two animals and surgically connect them to the point where they become essentially like conjoined twins. So what happens is they get surgically connected just at the skin, and they develop spontaneously vascular connections between them. So we don't do any vascular surgery. We just connect them under the skin. They spontaneously develop these connections, and within a couple of weeks, they have a single shared circulatory system. Okay, so the tissues of one animal are being bathed by the blood partially from the other animal. Okay, so we did these kind of control experiments, which we call isochronic, the same age, where we paired two young animals together or two old animals together. But of course, the interesting experiment are the so-called heterochronic pairs, young and old, a young animal to an old animal paired. So we did this, and we then kept these mice, and we've done this many times. We kept these mice together for 2 months, first time we did this. And then we injured the muscle of the old animal, and we asked, what happens then when you have an old tissue that has now been bathed in the circulating environment of this young animal for a couple of months? What happens to the regenerative capacity of that tissue? So if we look at the isochronic pairs, here is normal muscle. Here's an area of injury that's in the process of repairing. You see a lot of these new muscle fibers being formed, just like an unpaired young animal. If we look at the old isochronic pairs, two old animals paired together, here's the uninjured muscle looking normal, and here's the area of injury where you see a little bit of new muscle formation, a lot of scar tissue being formed. But if we look at the heterochronic animals, the old pair, we see really what looks like normal regeneration from a young animal. So uninjured muscle here, and a lot of very, very healthy regeneration going on in this otherwise older mouse. So this was, again, a kind of an extension of that previous experiment, but what we found was that this was true across many tissues. So if we looked at regeneration in liver, it was enhanced in the old animal. If we looked at neurogenesis in the brain, it was enhanced. If we looked at hematopoiesis in the blood, it was enhanced. So clearly this was not something that was limited to muscle, and clearly this phenomenon of stem cell aging, we think, is related to an age-related suppression of stem cell activity that is relieved in the setting of these heterochromic parabiotic pairs. Now, there's no free lunch, so the young animal had to give up something. And indeed, if we look at the regenerative response in the young pairs of these heterochromic parabiotic pairs, regeneration in every case was impaired. Okay? So what we've really focused on now is looking at the age-related changes that occur in the blood and in tissues that we think are actually suppressing stem cell function with age and are being either diluted or relieved by these heterochromic parabiotic pairings. So we're beginning to get to this idea that there are these age-related changes in protein levels, some go up, some go down, that are actually regulating stem cell function generically across many, many tissues. Yeah?
Here potentially you have two sets of genes active, like two mouse you join them, you may have two different sets of genes.
But these are inbred strains of mice. You mean, so the question is, are the mice genetic? The mice are genetically identical. Okay, sorry. So the question is, pairing two mice. Obviously, if we pair two mice that are not genetically identical, we'd have problems with rejection. We'd have to deal with that. But these are genetically identical mice. So yes.
Did you disconnect the mice, and if so, was there lasting effect?
Okay, excellent question.
Your mouse?
So the question was, have we disconnected these mice and looked for the loss of these effects? And in fact, we have. And interestingly, the effects last for quite a long time, meaning if we've kept them together for 2 months and we disconnect them, 2 weeks later and even a month later, we can see some declining but lingering effects. So it's not an instantaneous loss of this effect when we disconnect them, but there's some memory. We think there's some actually reprogramming of the stem cells that has a lasting effect and takes some time to wear off. Yes?
How profound is the effect on the younger mouse, the negative effect on the younger mouse, and how long lasting is that?
So the question is, how profound is the negative effect on the younger animal, and how long does it last?
How long does that last after the disconnection?
So we haven't done, and how long does it last after the disconnection? We haven't done too many of these disconnection experiments, so I don't have too much information on the time course. But we certainly have looked at the impaired regenerative response in the young animal of these heterochromic pairs. And depending on the tissue we look at, a little bit depends on how you measure it, the effect is profound. If we look at the effect on new neuron formation in the brain, the effect is really quite dramatic. Okay? It's less dramatic in the skeletal muscle stem cell compartment. So it depends a little bit on the tissue, but the effect is, again, I think is quite profound in places, in particular in tissues where the normal age-related changes are quite dramatic. Yes?
Is this effect seen in blood transfusions?
Okay, so the question is this effect seen in blood transfusions? So to my knowledge, no one's ever looked, and we've looked to see if anyone's looked. A lot of blood is not coded for age, so it's hard to find out if that's true. Remember, these animals are connected 24/7 for 2 months. So I often get asked that question of, did we do this by a blood transfusion? We never did it because I never thought it would work. But it turns out our collaborator, Tony Weiss-Corey, faculty member here, actually his graduate student, Saul Vieta, did an experiment after having done this heterochromic parabiosis and looking at neurogenesis, so enhanced neurogenesis in the old animal, suppressed neurogenesis in the young animal. He took blood from old animals and did a series of transfusions and saw he saw less of a profound effect, but saw the same direction of effect, a suppression of neurogenesis from blood transfusions. Now, he did quite a few of them over a short period of time, so it wouldn't, again, be analogous to what we see, what might be occurring in humans from a single transfusion. But it's an interesting idea that what an effective transfusion might be more than just the blood itself. Okay, so really this gets to us this fundamental question, I think, of is aging reversible? And this is a painting of the Fountain of Youth by Lucas Grenache the Elder from the Weimar Republic in the 16th century. And this is a painting in which he has, for some reason, it's all women being carted in on the left I don't know why. And what's that? Yeah, maybe, that's right. And it's, of course, they're coming in from this very bleak landscape, okay, where everything is stark and harsh. Being examined by some physicians, they enter the fountain of youth, and they emerge as nubile young women. And they're clothed, and they enjoy all the fruits of youth, romance and sex and good food and all the great. So this was his idea of the fountain of youth. And you know, for us, the question is, are we beginning to understand something about aging that is in fact reversible? And we don't know what the total consequences of reversing this phenotype is. We don't really understand the mechanism yet, but clearly, from the point of view of just asking, can you have old tissues restored to youthful capacity, the answer seems to be yes. Now, I can imagine that if one did this chronically, many things would happen that would be untoward and undesirable. But for these kind of, the way I see this thinking therapeutically is not to reverse aging or to revert or to stop aging, but actually to help with repair of injuries in older individuals. So can we take a bone fracture and help it heal more rapidly and more effectively in an older individual? A very limited treatment in terms of time and in terms of space. I think perhaps it's possible we could get away with that without having the untoward effects of stimulating stem cells in older people, which I think one of the greatest risks would be the development of cancer. So, you know, we want to stimulate cells at a very specific time in a very specific place and see if we could get away with that to enhance tissue repair without having negative consequences. Yes?
How does your research relate to telomere research for aging?
So the question is, how does our research relate to telomere research in aging? I mean, it's an excellent question. So in the field of aging, of course, the issue of telomeres is as cells divide over and over, the telomeres shorten, and ultimately there's a potential for the cells to be able to lose their ability to divide. So they die or they stop dividing because their telomeres shorten. We haven't actually looked at telomere length in these cells, but they don't, as far as we know, express this enzyme called telomerase, which you would need to restore telomere length. So if that were the mechanism by which this were working, these cells, we think, would have to express this enzyme so that the old cells, which have shorter telomeres, would then be able to have longer, quote, younger telomeres. We don't think that's happening. Okay? So we don't think this is related to telomere biology. But that's a good question.
Do you want to comment on the experiment of endurance athletes in their 40s and 50s and the effect that they have longer telomeres?
So Phil was just mentioning the idea that endurance athletes appear to have longer telomeres. Are you? Yeah. So I mean, I just know the phenomenon. I mean, I don't know if anybody knows the mechanism of that. Yeah.
There is a series of studies that at least demonstrate.
Right. So the question is, are they? So again, the comment is with endurance athletes appearing to have sustained longer telomeres than their say control athletes or control individuals. And the question is whether this is because through endurance training they're just not shortening their telomeres as fast, or they're actually expressing telomerase and lengthening their telomeres. So either way, it seems like a good thing, even if we don't understand the mechanism. Yes?
The other way around, the fact that they've got longer telomeres makes them better endurance athletes.
I'll turn that over to Phil. Actually, I mean, you know, to my knowledge, I don't know. I mean, I think we, as far as I know, there's no reason to think that athletic ability is limited by telomeres that I'm aware of. So I would predict the answer will be no, but I'm speaking more from speculation than from knowledge. Okay, so this is where we are in terms of thinking about this kind of intersection, interesting between biology of aging and the biology of stem cells. I think they come together very interestingly here with potentially kind of a therapeutic angle that if we begin to understand what makes a stem cell age, its environment, and how we can change that environment to enhance the function of stem cells, we think we'll learn some basic biology and also potentially have some therapeutic window there. So I just want to end now with just some food for thought that, as I was putting this together, that I think are interesting questions to think about both from a biological point of view and from a societal point of view, is that if we could extend lifespan, should we? I mean, this obviously is a philosophical question that I think is an important question for society to answer. Should we be investing in research to extend lifespan? I think most people agree that research to extend health span is a good thing, but is it worth it as a society? Should we be having people striving to live longer maximally as opposed to live healthier for the amount of time we have? What are the societal implications of this rectangularization of the life curve? I mean, if we really can start doing this, if we really could start taking care of the diseases of aging, what would that mean for society as we have now, where people retire in their 60s, and what if they could live to be 120 and quite healthy? What would they do for the next 60 years of their life? How would we change? How would society deal with that? And then I know you talked about this last time, is that what are the cultural, the current trends, cultural or otherwise, that may actually be actually leading us to lose some of this great benefit we've obtained in the last 100 years in the extension of average life expectancy. I mean, clearly the obesity epidemic is actually threatening to have, for the first time in 100 years, to have us actually have a shorter average life expectancy. From all of the advances we've made, it doesn't mean that it's going to continue just because we've made these progress so far. So this is my group from my lab. I just wanted to thank them for everything we've done is their hard work and their creativity. And I'll just stop there and take any other questions before I turn it over to Anne. Thank you very much.
All right, so good evening, everyone. And it's a real pleasure to be here tonight to speak about the topic of interest tonight, which are the mechanisms of aging and longevity, and to pursue in the theme that Tom already introduced and spoke about. So as you can see on this drawing from Leonardo da Vinci, there are obvious differences, outward differences, between a young adult and an old adult. And those outward differences are also accompanied with a decline in a number of physiological functions that affect a number of different tissues and organs in the body. And for example, if we take the brain as an example, there is a decline as early as in the 20s and the 30s, this slow but progressive decline in a number of cognitive functions, including working memory, short-term, long-term memory, speed of processing, although you can notice that verbal knowledge stays pretty constant, but it's probably due because of the fact that people get exposed to much more words as they get older. Now, this decline in a number of physiological functions, in a number of tissues, including the brain, is also accompanied by the increasing onset, strikingly, of a wide range, a wide spectrum of age-related diseases. So you can see, as humans get older, this striking increase of diseases such as heart disease, cancer, stroke, emphysema, type 2 diabetes, neurodegenerative diseases such as Alzheimer's, Parkinson's, etc. So an important question at this stage is aging itself a regulated process? And could maybe understanding aging in and of itself help us understand a common mechanism behind all these age-related diseases and help us identify maybe new avenues to treat and prevent those diseases instead of treating them one by one, finding the root cause or the root mechanism behind all those diseases. Now, for the longest time, aging was actually thought to not be a regulated process, to just, as Tom was mentioning, be just wear and tear or the rusting of the organisms. But actually, this view has changed, and it is really now thought by experiments, seminal experiments in the last decades or so, that aging is actually a very regulated process, and that it can be regulated by a combination of genetic and environmental factors. And for example, studies in twins in humans have indicated that the contribution of genetic factors can be as much as 25 to 30% to human longevity. So in the next slides, couple of slides, I'm going to present examples of how genes and how the environment can regulate aging. And obviously, an interesting aspect is also how what happens at the interface between genetic and environmental factors and how gene products can really integrate environmental changes to, in the end, affect lifespan. An indication that aging and longevity are regulated by genes came, as Tom was already mentioning, from observation, genetic studies in humans with families like this family here, which is an American family repatriated in Utah, where both parents were centenarians, and 8 out of their 10 children were also centenarians or living more than 95 years old. Studies in families like that by the group of Tompers have revealed regions in the human genome, on human chromosomes, that are associated with this exceptional longevity, being a centenarian. So there are, for example, regions on chromosome 4 that are in human that are associated with exceptional longevity. But so far, those studies haven't been able to zoom in and pinpoint the specific genes that are on those chromosomal regions that are responsible for this exceptional longevity in human. And what really made a breakthrough, a key breakthrough in the world of aging to identify the specific genes that were important for regulating lifespan was studies in a very tiny 1 millimeter worm called Caenorhabditis elegans. So it's a little tiny worm that lives in the soil. And experiments by the group of Cynthia Kenyon at UCSF back in 1993 identified worm mutants that could live 2 to 3 times longer than wild-type normal worms. And not only could those mutant worms live longer, but they were also more youthful as far as worms were concerned. So they were more active, they had less wrinkles. So they kept their, it's not that their decline period was extended, it was really their youthful period of life that was extended, both their mean and maximal lifespan. And it turned out that those worm mutants that can live 2 to 3 times longer had a mutation in one single gene, a gene that encodes the receptor for insulin. Now, this was revolutionary as well because people had already a hard time realizing that aging could be regulated by genes, but let alone a single gene. It was thought to be very complex, and maybe that 10 or 100 genes would be needed to affect lifespan. But here, that really said that, at least in a worm, mutating a single gene was enough to extend the youthful period of the worm by 2 to 3-fold. Now, this mutation in the insulin receptor that extends lifespan in a worm turns out not to be an idiosyncrasy of this tiny round worm in the soil. And it actually turned out by further experiments done in the fly, Drosophila, in mice, and now even in human centenarians. Now that we knew the gene, it's easier to go back in those exceptional centenarians. And they also, some of them turn out to have the same similar mutation in this insulin receptor, and indicating that this insulin mutation that lower the activity of the insulin receptor in a conserved manner from worms to human have this ability to extend longevity, or to be associated, at least in human, with extension of longevity. Now, how does that work? How does, at the molecular level in the cell, how does reduction of activity of a receptor, the insulin receptor, how does that act? Well, normally, insulin is a circulating hormone that, for example, gets circulated in the blood when we eat a meal. And this hormone will bind to receptors that are present at the outer surface of the cells that contains this receptor. And what this triggers is a cascade of events, signaling event, which culminate in what's called the phosphorylation, which just means the modification of a family of proteins in the cells, called the FOXO transcription factors, and it modifies them and it prevents them from acting. Normally, those factors would be acting to regulate a lot of genes in the cells, and in this case, they cannot no longer act. And those are conditions that are associated with aging. Now, in those worms that live very long, or in those mice or flies or even human that have lowered mutation in the insulin receptor, or if one has lower amount of circulating insulin, this whole pathway is no longer set in motion. And what that means is that this factor that was normally kept inactive now becomes active. It can go into the nucleus and it can activate many, many genes, and those genes form a program a program that is important in the cell to detoxify those reactive oxygen species that Tom talked about, and to repair a number, to enhance the repair process of the cells. And those are conditions that are associated with longevity. Yeah. So there is less receptor in this case. If there is absolutely, this is an oversimplification. If there is absolutely no receptor, this is really bad, and there is actually no organism that gets alive because it's lethal. So the mutation is that's why I said reduction in function. It's not a total loss of function, yeah. So the question was, was there no receptor at all? And in fact, those worms still, or those humans still have a receptor, it's just less active than the normal one.
And these receptors are active like right from the birth, or they get manufactured?
So are those receptors active right from the birth? From birth, and indeed they are active right from birth, but then they play a slightly different function during development. They're actually very important for the growth of an organism, and it's actually why it's really needed to have them. But then, as one becomes an adult, they are not as beneficial anymore, and they actually turn up, what's good for growth during development turn up to be detrimental for an adult aging. All right. In the past couple of years, it has become clear that, again, in human, this pathway that I just talked about is really conserved, and what was discovered in the tiny worms has hold true now for humans that live actually 2,000 times longer. We live 2,000 times longer than those miniature worms that live like 15 days or so. And yet, similar in papers that were just published, if one has a specific type of those FOXO molecules that are downstream of this insulin pathway, one is more likely actually to, or has more risk, or if you prefer, like more chances to live longer. And if one, so those mutations are actually quite common, and nowadays one can even like find out if one has this type of FOXO in their genome, like this type of mutation, that type of mutation, or that type of mutation. Those are the different possibilities. And if one has that type that's associated with just regular lifespan, one lives 70 to 80 years, if one has those type of mutation in that specific gene, this is associated with possibility, increased chances of being a centenarian or even a super centenarian. So this indicates that this discovery made in a worm has actually revealed, like this is published like last year in 2009. So it's taken a while to go from the worm to human, but it is indicating that there is conservation in the pathway that can regulate lifespan, and that studying model organism can have a huge impact in our understanding of what are the genes that regulate lifespan in human. So this was for genes. Now, what about the environment in this? So obviously, there are lots of ways in which the environment can be detrimental for lifespan. For example, smoking is detrimental for lifespan, pollution, etc. But there is one environmental intervention that's extremely beneficial for lifespan, and Tom already alluded to it, and also Phil. And it's called dietary restriction. So dietary restriction is defined as restriction in food intake without malnutrition, so supplemented in vitamins and minerals, for example. And you can see here in experiments done in mice by Richard Weinrooks that restricting food intake by 25 to even in this case 60% extends both the mean and, surprisingly in fact, the maximal lifespan of those organisms. And dietary restriction is not only working in mice to extend drastically by 60 to 80% mice lifespan, it's actually an intervention that can extend lifespan in virtually all species that have been tested so far. So people have done experiments on spiders, on trout, on dogs, on the model organism that we use in the lab, yeast, worm, fly, and mice, even on primates. This seems to be working, and there are actually now humans who self-report to perform dietary restriction, and they self-report to have health benefits. We don't know about their longevity yet, but health benefits that are associated with increased in longevity. Now, dietary restriction extends lifespan, but interestingly, it also delays a number of age-related traits and age-related diseases. So again, it's not something that just prolongs misery and just extends the decline periods of life. It has been shown to decrease the risks of cancer in both mice and human. And it also has been shown, interestingly, to slow the decline in learning and memory. Part of this decline that I showed you in human that occurs, it also occurs in mice and in rodents, and you can slow that decline by applying dietary restriction. And here is just to show that in the presence of dietary restriction, a region in the brain called the hippocampus that's really key for the formation and the storage of memories is really preserved, whereas in a usual diet, you can see that there is a lot of already death and degeneration in that area. So dietary restriction has this amazing impact in all animal kingdom, including primates and probably human, to prolong lifespan and prolong health span. There is an evolutionary theory of why would that be, that dietary restriction is so potent at extending lifespan in such a conserved manner. And the evolutionary theory goes as follows, that in times of food scarcity, dietary restriction, this mechanism, food scarcity, would extend lifespan such that the individual can allocate the little energy that they have to the maintenance of the youthfulness so that they can reproduce, but only later on when better times become available and reproduction, the survival of the youth is more ensured. That's just a theory, but that may be explaining why it has such a pervasive impact on lifespan in so many different species. So because of this amazing ability of dietary restriction to extend lifespan and health span, one can ask a key question, which are, what are the molecular mechanisms by which dietary restriction or caloric restriction can extend lifespan? And if we understand those mechanisms, can we harness the benefits of caloric restriction without necessarily having to perform caloric restriction, which actually is, like this, like 40% to 60% reduction is actually very hard to do. That would be the equivalent of 1,200 calories per day, which is very hard to make it compatible with an active lifestyle, for example. So can we understand how it works, harness the benefits of it, maybe mimic it by some, let's say, drugs, at least mimic some aspects of it? So here is the question. Now, so this is rephrased it more in a, can we understand what's this black box here and how dietary restriction, diverse way of doing dietary restriction, how do they extend longevity? Yeah.
Does the dietary restriction have the same effect no matter when it occurs in life? Or is it, if you are very thin when you're young, it extends your life, but if you're thin when you're old, it doesn't?
Yeah, so the question is, does dietary restriction have the same impact no matter when it started in life? And the answer is no, but it does always have an impact. But it's not the percentage lifespan extension that it has is lower and lower as much as shorter and shorter the dietary restriction time is done. So if dietary restriction is started in young organism, this will have a huge impact on its lifespan. If it started at midlife, let's say, it will still work and still extend lifespan, but by a shorter percentage, and etc. So it's still like, if one is thinking about it, it will still be beneficial to start it, but the impact will probably be lower. Yeah. So one way by which one can connect the dots between what I said at the beginning about genes that regulate lifespan and environment, this amazing environmental intervention to extend lifespan, is a possible simple linear relationship which goes as follows. If one diet does caloric restriction or dietary restriction, it's like eating less. And as I said, insulin is this hormone that one gets when one eats a meal. So if one eats lower amount of food, one will probably have less circulating insulin, and in fact that's the case. Rodents and humans that are on caloric restriction have lowered amount of circulating insulin. Therefore, this pathway that I was telling you about is going to be less active. It's going to be sort of like those mutants that have a reduction in function in their activity. So it's mimicking those insulin mutants that I talked about. That means that their transcription factor, their FOXO, is going to be mostly active and in the nucleus, which is good. It's going to trigger all those genes that are repair, detoxification genes, and stress resistance type of genes, and that may lead to longevity. Yeah? Yes. So this is a very good question. So in diabetes, you may have all heard that people who have type 1 diabetes don't have any more of those cells that produce the insulin. They destroy them, so they cannot produce the insulin any longer. And in type 2 diabetes, it's slightly different. People start getting resistance. They have actually increased amount of insulin circulating, probably as a compensatory mechanism, but they start being resistant to insulin. So certainly, tweaking this pathway too much in the other direction, lowering this pathway too much or completely, can be very detrimental. Again, this can lead to complete death, and in some cases, diabetes. But I think here, the interesting part about those worms, and I think this is something to think about, one can think about of this pathway as, okay, let's just lowering it right enough to be in the right window, not too much, then it's death or diabetes. One can also think about it in that way, but adding on top of that the layer of regulativity, meaning that what happens in diabetes is that this pathway is misregulated, so it's like either firing too high or firing too low. Those worms or those humans that have lowered their pathway for longevity, they have lower of it, but it's still nicely regulatable. So when insulin comes and goes after a meal, it's still doing its job, those waves of going in and out pretty well. So in fact, I think it's not teased out. This is a very good question, and I think a lot of researchers are working on those questions right now. And the idea would be that the exact amount of activity is important, but also the regulativity, the plasticity of it is also very important. Yeah.
Is there a relationship to weight itself? So, for example, if you had dietary restriction and you reduced your body weight, that's one way of doing it. But from a thermodynamic point of view, if you increased your activity and achieved that same weight, does that have an effect or not?
So the question is whether it's the weight or if it's the intake of calories, and if it's the weight, one could imagine that you could achieve the same effect by eating the same amount but increasing your exercise report. And I'm sad to report that it is the intake that seems to matter, and if animals are being exercised, they have a set amount of food, and they have exercised to the point that they have the same weight as the animals that are dietary restricted. They display health benefits of this, but this does not extend their maximal lifespan, whereas dietary restriction extends their maximal lifespan. It's not understood why. Some people think that exercise has some detrimental effect as well, that when summed up lead to, no, but it's an interesting point to contrast exercise and dietary restriction in that aspect.
Starving couch potato.
Exactly. Or take the drug that mimics caloric restriction and be a couch potato. Now, I'm not advocating that.
How did you identify the gene within the C out of the GONS that was FOXO3? How was that determined?
So so the question is how was FOXO identified? So FOXO was identified by many groups in many different ways, but the first characterization in the worms was by they had those mutants of the insulin receptor that lived very long, 2 to 3 times longer, and they wanted to know what was mediating that. So they did a screen for genes that, when mutated, could revert back the lifespan from very long to back to normal. And by doing that, the one gene that came over and over and over and over, the only gene that came up, was FOXO. So that's how it was identified genetically. And then molecularly, a lot of experiments went to characterize the molecule for what it was, meaning a nuclear transcription factor. Yeah. Yeah.
So what percentage of the centenarians have the mutation?
So the question is, what is the percentage of centenarians that have the mutation in FOXO? So it's limited. So which still goes back to the questions that there are probably many genes in the centenarian, in different centenarians have probably mutations in different pathway. So it's considered that the FOXO mutation accounted for 2% of the variation that they could see in, like a small percentage of the centenarians. Now, as you could notice, it's a whole pathway. So it's insulin, there's all these molecules in between, FOXO, there are actually different FOXOs, and I didn't talk about that. Then there are the target genes. So I think when all is said and done, it will be interesting to see what's the percentage of centenarians that have a mutation in that whole pathway. But FOXO in and of itself, it's actually relatively limited. Yeah.
Low glycemic index food, frequent meals, and get the same decreased insulin?
So the question is, can you vary the regimen, basically, so that you, for example, low glycemic index and lower the insulin and still have this extension of lifespan? To my knowledge, people are trying to do those experiments. For example, do the mice with the Atkins diets or like those kinds of different diets and then see in the end what happens. But the results, to my knowledge, the results are not known. Based on the research, I would imagine that everything that, well, not everything, but it depends, I guess. But things that will decrease the insulin levels, I think will turn out to be relatively beneficial, at least in terms of health span or mean lifespan. Maximal lifespan might be another story, and in this case, it might be really that it's the number of calorie period. All right, so that's one possibility. We have time, right? Yeah, so is it the only one, though? I mean, does everything revolves around insulin? Or as much as I would love that, because we work a lot on this, it doesn't seem that the insulin FOXO pathway is the only way by which dietary restriction acts to extend lifespan. And in fact, this is an experiment in worms. It's a genetic experiment, but what you can see in that experiment is that worms that live long because they have a mutation in the insulin receptor, in blue here, they can live even longer when they are put in a caloric restriction, in a dietary restriction regimen. So if the only thing that dietary restriction was doing was to reduce, to act on the insulin pathway, then it wouldn't be additive onto the mutation that already mimics that. So what that meant to a lot of people, those seminal experiments, what they meant was that, yeah, insulin, reducing insulin is good for extending lifespan, and reducing insulin is probably one of the ways by which dietary restriction extends lifespan, but it's probably not the only way, and there's probably other way in which one can extend lifespan downstream of dietary restriction. So what is it? So in our lab, we had this candidate approach. So we had this hypothesis that maybe there would be another system to sense calories, in addition to insulin, which is a very hormonal way of sensing food, right? You eat a meal, boom, you have insulin, and this senses that you've eaten a meal. Maybe there would be a more, in fact, ancient way that cells would sense really in a very ancient, evolutionary speaking, in a very ancient manner, could sense calories or amount of nutrients that there is. And in fact, there is such a molecule in the cell that's a very ancient, meaning it exists already in organisms that don't even have insulin, like yeast, for example. And it's present from yeast that don't have insulin to human, which do have insulin. So it's a protein that can sense energy in a different way. What is it? It's called AMP, activated protein kinase, or AMP kinase. And what's interesting about this molecule, it's a molecule in the cell, is that it can sense a wide range of stimuli. And it turns out that a lot of those stimuli are associated with dietary restriction. Low glucose, low energy amounts, low leptin, it's a hormone that's produced by fat, so low fat content. Interestingly, it's also activated by other types of stimuli, like exercise, as well as by two anti-diabetic drugs, fenformin and metformin, that don't act through the insulin pathway, drugs that act in another way than the insulin for diabetic patients. How does it sense all this? Those are a wide range of stimuli, and I said it's this very ancient molecule that can sense, count calories in another way than this insulin hormone. Well, it does that because it can exquisitely sense the amount of AMP, the ratio in cells between AMP and ATP. And ATP is energy. This is a molecule that contains a lot of energy and that will provide energy for most of the functions in the cells that require energy. AMP is the precursor of ATP, so it's like low energy. So each time in the cells that the ratio between low energy to high energy increases, this molecule gets activated because it can directly bind AMP. So it's really a molecule, it's a protein kinase. It can sense low energy very directly in every single cell in the body. And this AMP kinase was well known. It's, as I said, it's present even in yeast. And it's very well known to regulate very important processes in the body, such as lipid metabolism and protein synthesis. But a key question is, does this fuel gauge in the cells, this counting calorie molecule, can it mediate the longevity effect of dietary restriction? So how did we do that? Well, we used this little worm, this nematode 1 millimeter soil worm, Caenorhabditis elegans. And we devised a method of dietary restriction, caloric restriction in the worms. So they normally are on plates, they crawl around and they eat bacteria. And what we did is that we put them on their plates, and then at some point, we either give them all their food, like we would do for human or mice, or we gave them a third or half, or we restricted their food access. So when we did that, the worms eat less. You can see that here. And importantly, like in any organism, the worm that eat less, they live longer, both in terms of mean and maximum lifespan. They eat less, they live longer, and their AMP, not surprisingly, they eat less, they have less energy, so their AMP to ATP ratio increases because, and that's telling the animal, look, you know, I have low energy, my AMP is high, my ATP is low. So that means that probably this AMP kinase gets activated. Well, is it important? So for this, we can use a mutant in these little worms that doesn't have AMP kinase anymore. Wild-type normal worms, they live longer if you restrict their food. Mutants that don't have AMP kinase can no longer live longer in response to this dietary restriction. Presented in another way, you can see that wild-type worms fed replete ad libitum, they live a certain amount, 15 days. If you restrict their food, they live longer, and then boom, they'll die, they'll starve. So they'll live longer here for dietary restriction, and they starve. The mutant, it doesn't matter. Like, they don't have this energy sensing mechanism anymore. It doesn't matter what you do to them. They have this flat lifespan. They can no longer sense the dietary restriction. Now, dietary lifespan is one measure, but, you know, again, who cares if one makes the worm live long, but the worm is really, like, sick, and you just, like, basically make it, like, not enjoy life. So, in fact, we measured what we thought was, it's only a measure of worm, what we think about worm usefulness, is that we measured its activity, so its ability to have rapid movement in the plate and be relatively happy, at least we hope. And what we found was that during aging, there is a very striking decline in activity, in worm activity. It gets slower and slower. As expected, this is slowed down. The slowing of the worm is reverted by dietary restriction. Again, this amazing intervention to slow down aging. And again, the presence of AMP kinase, this energy sensing molecule, is necessary not only for the lifespan, but also for the health span of these little worms. So what that tells us is that one way by which dietary restriction extends lifespan and health span, it's not just by acting on a hormone that circulates in the organism, it's also acting in every single cell by acting on this molecule that can really sense how much energy there is to promote longevity and, to some extent, fitness. Now, yeah?
How do you count the calorie? Because when you buy food, they say 120 calories, how many sugar. Where those measurements coming from? Do you trust those calories?
Yeah, so the question is, like, on those little package, they tell you, like, how many calories there is in there, and where does that come from? I think it comes from a lot of measurements that have been done where, like, the calorimetry type of experiments where they measure exactly how much a lipid, a sugar, or a protein counts in terms of calories, and then they see what the food is composed with, and then, so I think it's relatively trustable what's on the label. Now, the type, so I guess, like, is your question getting at which type of, like, is sugar, lipid, proteins, would they all need to be decreased? Like, can we just decrease sugar, for example, and be fine in terms of proteins and lipids, or do we have to decrease certain types of calories, let's say coming from fat or coming from sugar? It's not completely clear yet 100%, but it seems that based on studies in model organisms that in fact proteins matter a lot in flies, for example. So amino acids are very important to restrict. So, you know, like those Atkins diet, for example, would not be recommended for extending lifespan because it seems that one needs to restrict amino acid, and that's very important in the ability to extend lifespan. Now, in mammals, the type of calories, the type of, you know, whether it's proteins in the diet, lipids in the diet, or sugar in the diet, it's not clear. It seems to actually, in mammals, if anything, just due to the total amount of calories. And so far, in the experiments that have been done, it doesn't seem to matter at all where the calories come from. But I think this is an important question that has yet to be solved.
Can we do ourselves? Is there any formula we can just download from somewhere and then we eat it and just calculate it?
Yeah, so can we use a formula to calculate that? People who are doing the self-reported calorie restriction are called, some of them are in a group called the Cronies, C-R-O-N-I-E-S. And the Cronies have a website where they explain, and the head of the Cronies Society is this person, Paul McLaughlin, where he has made the calculation of what it represents for a man or for a woman, which is, it's different actually, and based on the level of activity, based on the age, what is the amount of calories that is required, that would be required, and how to calculate that. So this website is actually quite useful if someone wants to implement those kinds of things, yeah. Yeah.
How would the extreme restriction of calorie affect women's ability to reproduce?
Yeah, so the question is, how does restriction of calories affect women's ability to reproduce? So that's a very good question as well. Obviously, it's like what Tom was saying, there is no free lunch, meaning that, well, in that case, there is no free lunch, in the sense that basically, when one restricts calories, this has a negative impact on fertility, especially female fertility. So females that restrict their calories during their reproductive time have either inability to be fertile or less ability to be fertile. Now, they also, not paradoxically, but because they extend their lifespan, they will still extend their reproductive time also. So there is a lower fertility at each point, but the fertility period is extended. And as I was mentioning to the question that was asked, caloric restriction can also be started later in life. So one could imagine starting the caloric restriction regimen if one wanted to extend their lifespan past the reproductive time.
I would assume you would not do extreme calorie restriction on a child.
Yeah, so the question was, wouldn't want to do extreme caloric restriction on a child, and that's true too, because again, it would have a very negative impact on growth, and then growth would be stunted, and so one wouldn't want to do that. But because of this ability to start in model organisms as well as in human caloric restriction later in life and still have very important benefits, it's still possible to imagine windows of time whereby one would do that are past those critical periods, yeah. Yeah, in the back.
So if one began calorie restriction, say, when a child had grown to adulthood, and then stopped for a while, have kids, and then started up again, are the effects cumulative?
So the question is, can we do the caloric restriction at different times, do it, then stop doing it, and then is the effect cumulative? People haven't done good design like this, but they're starting to do that, and it does seem that the effect is cumulative in the sense that the longer you do it, it doesn't really matter exactly when, but the overall time is what matters in terms of the lifespan. Yeah. Yeah.
Do people who do this feel good?
So yeah. So the question is, does it make you live longer, or does it make you feel that you're living longer because the life seems very long? Actually, do people feel good by doing that? So there are two different camps on this. The first camp, the Crooney Society head by Paul McLaughlin, he raves about it. So he says that he's been able to play chess tournament and he feels really energetic. Actually, the mice that are placed on caloric restriction and the primates, they are less lethargic. Like they go, like they seem to be relatively active. So that's the optimistic camp. And then there was this experiment that was done in Biosphere 1, like, where they were the biodome. And in fact, essentially, the people that went in there, because they actually screwed up their crops the first year, they actually essentially were caloric restricted in there. So they had really good blood parameters when they came out of this biodome experience. There are papers on that. They had really good blood markers, and probably they have beneficial impact on lifespan, but psychologically, they were followed by, maybe it was also the confinement, but the restriction on food on some people had a really marked, very negative psychological impact. So some people fear that caloric restriction has a very, very negative on human psychological impact. So those are the two camps right now. Yeah.
Is increased lifespan also associated with decrease in things like heart disease, strokes, cancer, those kind of things?
Yeah, so is increased lifespan, especially in response to dietary restriction, associated with health benefits, especially for diseases such as stroke, cancer, or all sorts of diseases? And actually, yes. So for all the models so far that have been tested of disease, dietary restriction has a very positive impact on those models. Yeah?
Just curious, I can imagine that every pharmaceutical company is trying to target insulin receptors and
Yeah. Yeah, so the question is about, and I guess we probably, I'll address this question by probably just wrapping up, and then maybe we can open the final discussion to the thing. So I was going to talk about specific mechanism, but I'm just going to, those are more like the thing that we do in our lab, and actually I put reviews on the web so you can see exactly all those specific mechanisms. I guess I was being ambitious here. So I'll all right, so to address your question about drugs, and coming back to the end, can we mimic those? So all the parts that you have missed, quote unquote, you can find on those reviews on the web, and there are more details about the mechanism, but you've seen the main point, which is that this AMP kinase, can this be important? And there are actually activators of AMP kinase in this French like here, those drugs, fenformin and metformin, and they can extend, indeed, lifespan, at least in rodent model. And there are also other molecules, such as the resveratrol present in red wine, Activate 31, another molecule, but also AMP kinase has some impact, at least in obese model for rodent. And then finally, another molecule that's also in the same pathway as AMP kinase called rapamycin, just shown this year, like very fresh result, showing that even in midlife, started in midlife, has a very significant impact. It seems small, but it started at midlife of mice. Very interesting increase in lifespan. So yeah, there are potential drugs that can potentially mimic dietary restriction. Can they be used? Can they be safe? Can they be combined to extend even further the lifespan? Those are all very interesting questions. So with that, I guess I will just close by thanking people in my lab and thanking you guys for being a terrific questions, and this was great.
Well, that was fantastic. Here's the CR Society if you want to look it up. It's right there. It looks interesting, but maybe if you want to drink wine, I think you have to have quite a lot in order to have the effect. So go do that, but do it after you get home. So I would say this has been a fascinating discussion and interesting to me personally because my mother is celebrating her 90th birthday today. So I don't know whether that's a good or a bad thing. For her, it's a good thing, but we'll see. More years of me may not be a good thing. But with that, thank you all for being here, and thank you, Ann and Tom. Great.
For more, please visit us at stanford.edu.